Okay, so today we're going to be going over videos 3 and 4 from this week's material, which was video 3 was on the cell cycle checkpoints and video 4 was on cancer genetics. Okay. So starting with the G1 checkpoint or the restriction point, this is the point of no return in the cell cycle. So just to recap, G1 phase is when the cell creates all of the necessary regulatory proteins and checks that it's prepared to synthesize the new DNA during S phase. It's when the cell grows, it makes sure that it has enough nutrients and things like that. And so it's making sure to see if cell division is viable. So in order to go into S phase, the cell has to make sure that it's ready to commit, or otherwise it will go into G0 phase. And so this checkpoint involves the activation of E2F. So if the cell decides that it does have all of the necessary requirements in order to pass into S phase, what will happen is that the inhibitory phosphate will be removed from CDK. It has to have the activating phosphate on it from CAC, and then the cyclin CDK complex will phosphorylate RB. This will result in RB being releasing E2F, and then E2F will be active. That's a transcription factor that will then be able to activate transcription of all the different genes necessary for expression of proteins involved in S phase. So the activation of E2F is the restriction point. Once this happens, the cell is committed to completing the cell cycle. So the next checkpoint, Laura doesn't really talk about this in her video at all, so I'm just going to go through it really quickly, is the G2 phase checkpoint. This is when the cell makes sure that the DNA was replicated properly and that it's ready to divide in M phase. And so the cell is essentially getting ready for chromosome segregation, and then they're checking for DNA damage and successful completion of DNA replication and making sure that the cells are big enough in order to divide. So during this checkpoint, what will happen is that M-cyclin CDK, if the cell passes the checkpoint, M-cyclin CDK will be phosphorylated by Wee1 and CAC, and then that inhibitory phosphate from Wee1 will be removed, very similar to the G1 cyclin CDK complex. And then once that phosphatase removes the inactivating phosphate, it will send the cells into M phase. I don't have the chat open, so I just want to make sure. Okay. The next checkpoint is the spindle assembly checkpoint. So this is making sure that each chromosome is attached to 2 kinetochore microtubules. So during M phase, you have the disassembly of the cell's microtubule kind of like system that it has set up in the cell. And then what happens is it generates the spindle through the centrosomes. And the spindle's assembly is, once the spindle assembles, it has to reach out across the cell and attach to 2 kinetochores on each of the sister chromatids. And the cell won't start pulling apart the sister chromatids until they have the proper microtubule attachments and the chromosomes are lined up at the metaphase plate. So there are a bunch of different proteins that are involved in this process. You have checkpoint proteins such as MAD and BUB, which are holding on to CDC20. Once the proper, oh, sorry, I thought you were like looking at the camera. Once the proper attachments have occurred, the MAD and BUB checkpoint proteins will release CDC20, which will then bind APC, and this will activate APC. So this diagram is showing it here. Once you have an active APC, this is a ubiquitin ligase. It can go about its function as an enzyme and ubiquitylate securin, and securin holds separase inactive. And so once securin has been ubiquitinated, it will be degraded. And now you have separase no longer being inactivated. So now separase can go on and perform its function, which is cleaving cohesin. And cohesin is the complex that's holding together the sister chromatids. Once cohesin has been cleaved, the cell enters anaphase and the sister chromatids are able to be separated. But none of this will occur unless the proper microtubule attachments are occurring on both of the kinetochores. And so that is the spindle assembly checkpoint. Okay, and so now the last thing Laura talked about in video 3 was P53. It's a transcription factor, which is and a central regulator of cell stress responses and is often involved in cell cycle checkpoints. So P53 becomes phosphorylated and activated in response to DNA damage. It will cause transcription of P21, and this is a type of CDK inhibitor. And so, for example, G1 cyclin CDK is inhibited by P21. So if you wanted to stop the cell from going through G1 phase, you would, by detecting DNA damage, you would have P21 activated by P53, and this would inhibit G1 cyclin CDK. And then P53 can also be involved in signaling for programmed cell death if it's determined that the cell cannot fix whatever's wrong. So if the DNA damage is kind of beyond repair, then the cell will undergo programmed cell death. And now we have just a couple of practice questions. And let me know if you guys have any questions about the material that I just went through so far. So the first question is a true or false, and it says DNA replication occurs immediately after passing the restriction point. And feel free to unmute or just drop it in the chat, whatever you think.
So they said false in the chat.
Yep. Yeah. So false. That's because G1 isn't over once the cell passes the restriction point. It still has some more growing to do. It still has to prepare for S phase. So it's not immediately ending G1 and going into S phase once it passes the restriction point. The next one is at what checkpoint are cells checking for proper kinetochore attachments to microtubules? Yep, yeah, that you can call it M phase checkpoint or spindle assembly checkpoint. They're synonymous, but yeah, the spindle assembly checkpoint is when you're checking for those proper kinetochore attachments. The next one is a bit longer. It's what would happen if you have a gain of function in P53? That's okay. I can go through it. So remember that P53 is a transcription factor that will activate
Go back to the slide.
Go back to the slide. Okay. Yeah.
That picture is helpful.
Yeah. Let me go back. Oh, here we go. Okay, so P53 is a transcription factor that activates transcription of P21. So if you have a gain of function in P53, that means that P53 can't be inhibited by anything. So here you see it's being held inactive by this MDM2 protein. And so if P53 has a gain of function, it will cause transcription of P21, which is a CDK inhibitor. And if you have overproduction of a CDK inhibitor, this will prevent the cell from being able to go through the cell cycle. And so most likely, if you have a gain of function in P53, the cell won't be able to go through the cell cycle, and this will likely lead to programmed cell death. Is gain of function always on? So no, gain of function would mean like if there's a mutation. So whenever we talk about like a gain of function or a loss of function, that's talking about a hypothetical scenario where, you know, essentially everything is turned on. What do you want to say?
So I think Ryan, I'm sorry.
Yeah, Ryan.
like the result of the, I think they're asking about the result again. So is the gain
Oh,
function
okay.
gain of
Yeah.
function?
So yeah, in terms of the question, if there's a gain of function in P53, that means it's always on. It's always causing activation of transcription of P21. Does that answer your question? Okay, cool.
One caveat that I want to add is that like just saying gain of function this is actually a really good question, because gain of function is relatively non-specific of a phrase, right? Do you want to go back to the P53
Yeah.
picture just as an example? Because you can have a gain of function here in about like 50 different ways, right? You can say like the P53 never lets go of the phosphate group, so it's always phosphorylated, so it's always going to want to attach to the DNA. So that might be one way you can have a gain of function. You can also have a gain of function in that once an activated P53 attaches to the DNA, it never comes off the DNA, right? So there's like multiple different points in this process and multiple different ways you can mutate P53 to induce a gain of function. In general, if you see something has a quote unquote GOF or gain of function, just assume that it means that it's always on, right? But like for the purpose of the problem set, the exams, things like that. But if on a question you have some specific gain of function, right? Like P53 is always phosphorylated, that you have to keep that in mind. That's the specificity of that gain of function in mind. That means that if P53 is always phosphorylated, it can attach to the DNA, right? One P53, once it's been activated, might attach to the DNA, and then later it might come off naturally. But then it still stays phosphorylated even after it's done its activity. So that one P53 can go back over and over and over, right? It's a slight caveat that that's different from if the P53 attaches to DNA and never comes off. Do you see what I'm saying? So you can have the gain of function, like multiple different types of gain of functions result in the effect being quote unquote always on.
Yeah, I think another example of that like kind of caveat would be in cyclin CDK. So let's say you have a gain of function in like the binding pocket for the WEE1 inhibiting phosphate. That would mean that like that phosphate maybe can't be removed by the phosphatase, and then you always have inactive CDK. So definitely it's very relative to what it's talking about, but in that question, and usually for the exams or something like that, it means that whatever it's referring to has like a constitutive on button, that it's always on. So yeah, hopefully that answers that question. So the next question is, what would happen if there was a loss of function in securin? So I'll go back to that diagram just so you guys can see it. So the question was asking if you have a loss of function in securin. And okay, I can go over it. Yeah. All right. So if you have a loss of function of securin, essentially there's nothing that's holding separase inactive, right? Securin is the molecule that holds separase inactive, that prevents separase from being able to go ahead and cleave cohesin. So let me just go back to that diagram. But essentially the answer that's written out here that you guys can always access on the slides is that like it would lead to cleaving cohesin before the cells may be ready, before there's the proper kinetochore attachments. And so that could lead to an improper separation of the sister chromatids. So if we look at this slide here, so you have securin, which is inhibiting the enzyme separase, right? And then when you have the proper alignment of all of your sister chromatids, you have securin becoming inactivated. And so that's through this whole process of, the MAD and BUB proteins releasing CDC20, which then binds to APC, which then ubiquitolates securin, and then securin gets degraded, so separase is active. that's a whole pathway that has to be gone through in order for the cell, once the cell recognizes that it has the proper kinetochore attachments at the metaphase plate. But if you have a loss of function mutation in securin, where it's perhaps no longer able to bind to separase, that means that nothing is inactivating separase. Separase can then go about cleaving cohesin even before the spindle assembly checkpoint is completely ready. And this can lead to improper separation of your sister chromatids. That can lead to aneuploidies, or, just non-disjunction in general, where you have the improper separation of your sister chromatids. Let me know if you guys have any questions about that. And I'm assuming Aman is getting to the question in the chat. Yeah. Okay. So those were all of the questions for the third video. Now we're going to move on to the cancer genetics, which is the fourth video.
Okay.
Let me just, yeah. Do you want me to?
What?
I have to stop sharing so that you can.
Oh, you want me to share?
Well, no, because you have the updated slides.
That's true.
Sorry, guys, just 1 second. Okay.
Yeah. All right, it's my turn. Okay, you guys can all see this. All right, so this is a figure that Laura had in her slides and in her videos. I like this because it shows like generally what is happening. So last week, you guys all learned about the signaling pathways and how the signaling pathways work, right? Now, that was like a general thing, like this is what a signaling pathway is. Now we're putting that into context, right? So what are some of those signaling pathways? Why are those signaling pathways important? A cell is always kind of existing in this state of flux, right? It has to like maintain its levels of everything, right? It has to maintain, if it's supposed to be alive, it has to like be alive. If it's supposed to carry out controlled cell death or apoptosis, it has to do that. The way this happens is the cell is constantly receiving signals, right? Based on the state of that cell itself and the state of the cells around them or elsewhere in the body. For example, if you have an infection, sometimes your immune system will tell certain cells to die if they're infected. That's like other cells around them telling them to die. Or if the example that Laura gave is if it comes off the extracellular matrix, if it's just like floating around in your body, it might be like, this is not where I'm supposed to be, so we'll carry out controlled cell death, right? So there are always these constant like positive cues and negative cues that are like kind of coming in, and the cell will balance that out. Ideally, for a healthy cell, that's where it's supposed to be and doing what it's supposed to be doing. The survival and proliferation cues, or the survival cues at least, should be balancing out or like overpowering the death cues and things like that. So if you have, so this is like what we were talking about earlier. If you have like a gain of function in those survival or proliferation cues, which means that they're always on, sometimes if the cell's not supposed to have those on, if the cell does come off the extracellular matrix or is in a place where it's not supposed to be, or it's not supposed to replicate, if you have a gain of function in those, that can lead to the cell replicating out of control, and that's cancer. Similarly, if you have a loss of function in the death cues or the no inhibition of growth cues, again, the cell won't die if it's supposed to, and it'll grow. It won't stop growing when it's supposed to stop growing, and it'll kind of grow out of control. Again, that leads to cancer. Does that make sense? Does the idea of like kind of the cell living in what's called homeostasis, does that make sense? Do you have anything in the chat?
Yeah, there's a question that says, what happens to cells that are programmed to die? Where do they end up?
Dead. Essentially, they, depending on how the cell, like what type of cell it is and um, what the situation is surrounding their death, most of the time they'll lyse, which is basically they'll pop, right? Remember that a cell has their the lipid like bilayer, right? Their cell membrane. A lot of time, if you put a hole in that cell membrane, it depends on the apoptotic pathway, right? The death cue, that with specific signaling pathway that we're talking about, like we learned last week, the specific signaling pathway will have specific effects. A lot of the time, the way I'll do that is just punch a hole in the cell membrane. And if there's a hole, that hole will expand and expand, and eventually the cell will just kind of like turn inside out. It's like if you pop a balloon, right? All set to move on?
Yeah.
Okay. So in regards to that, we're talking about like what leads to cancer here, these gain of function and survival and proliferation cues, and the loss of function, death and inhibition cues. And to get into that, we talk about these cues and the genes that are involved in those cues and the proteins in this language right here. That's the proto-oncogenes, which are genes that are normally in their normal expression and their normal function, they're used for the growth of cells, right? Laura used in her video, she used a metaphor of a car. I really like this metaphor, right? If there's a brick wall 100 feet down the road and that brick wall is cancer, then you want to be going at like a decent speed where you can slow down if you have to, right? So you don't want to be like really like slammed on the gas. You want to be in a lot of control. There we go. So that's what like proto-oncogenes are. Proto-oncogenes are like a controlled level of expression and normal function of these genes that are used for growth. If you have a mutation, does anyone know what kind of mutation this would be that would make them oncogenes, which would be like the foot is always slammed on the gas. So now you're like hurtling toward that brick wall. Is it gain of function or loss of function?
Someone said lost.
Okay. Actually, it would be the opposite, right? So if the proto-oncogene is the gas pedal, and that's used for growth, right? And if you're mutating it to become an oncogene, which is you're slamming on the gas now, right? It's a gain of function. Instead of pressing on the gas pedal lightly, you're really, really pressing it down. The cells are now growing. You're letting these proto-oncogenes instead of their normal capacity, they have a gain of function. So they're acting out of control. These proto-oncogenes are always on. They're always telling the cell to grow. If the cell is always being told to grow and it does grow out of control, that is cancer, right? Cancer is the uncontrolled growth of cells. Does that make sense? I don't know who it was that said that, but. Does that make sense? I'll use Hillary. Okay, I'm just gonna move on. In like the other thing that we talk about when we're talking about this control or tumor suppressors, and tumor suppressors kind of act as the brakes on this car. So the genes that actually regulate the proto-oncogenes make sure that the proto-oncogenes don't become oncogenes, and they're the ones that stop the cell from passing checkpoints, right? So if the proto-oncogene is pressing on the gas, then the tumor suppressor will pull that proto-oncogene back. It'll engage the brakes and it'll be like, wait a second, there's a brick wall ahead of us. We don't want to run into that. So let's not pass that checkpoint, right? In the cell cycle. A lot of times what they can do is they can incite, yes, the tumor suppressor is the brakes, yeah. They can like kind of, as part of this, they'll see that there's a brick wall coming up. So there's some DNA damage that might be a problem if this cell replicates, right? Okay, this cell is damaged. What do? Let's stop the cell in the cell cycle. Let's not let it pass this checkpoint, and let's fix that DNA damage, right? Or if this DNA damage is so bad, if the cell is so screwed that there's nothing, like it's past the point of fixing, right? Past a point of help, then let's cause apoptosis, control cell death. Let's sacrifice this cell for the greater good of the organism. Does that make sense? Okay, perfect. So just like a quick recap of P53, because P53 is very important in this context, P53 is a transcription factor for P21, which is a CDK inhibitor. CDI means CDK inhibitor, that's what the I is, and stops passage of the cell into the S phase, right? And P53 can also engage in apoptosis, engage the cell in apoptosis if there's some damage to the DNA that seems irreparable, right? So what is P53? Is P53 a proto-oncogene or a tumor suppressor? Right. The key thing is that it stops the passage into S at the G1 checkpoint, and it can engage apoptosis if there's significant damage to the DNA, right? If it's irreparable, if the cell is too screwed to be able to go on, right? That would make P53 a tumor suppressor, right? So if you have a loss of function, if P53 breaks, if a tumor suppressor in general breaks, the cell's going to be able to slide past that checkpoint and result in cancer.
What is apoptosis?
Apoptosis is controlled cell death. So how do I go back? So these death cues, right? That death itself for a cell is called apoptosis. The reason we have a special name for it is because the cell is doing that of its own accord, right? The cell is like, okay, I should die now, instead of a virus or something else coming in and making the cell die. The cell is seeing that there's some issue internally and causing a controlled cell death, and that's apoptosis. Perfect. Excellent. So if you have a loss of function in the tumor suppressor, if the tumor suppressor, in this case P53 is the example we're looking at, if the P53 breaks, the cell's going to slide by the checkpoint into S, and it's going to kind of run through that cell cycle much more easily and replicate much more easily, and you'll get out of control replication that results in cancer. So the key thing that we just essentially laid out is that there are two ways that you can get cancer, two main ways. One is gain of functions in proto-oncogenes, and the other one, that like those gain of functions make them oncogenes, and the other one is a loss of function in a tumor suppressor, right? So you can have a gain of function in proto-oncogenes, making them oncogenes, and that can cause cancer, or you can have a loss of function in tumor suppressors, and that can cause cancer. Keep in mind, one thing that I'm saying, and one very important word, is that it can cause cancer. Just because you have one proto-oncogene, right, or one tumor suppressor, let's say you have a loss of function in P53, the cell has so many tumor suppressors and so many proto-oncogenes that are constantly in flux, right? They're constantly kind of going against each other. The tumor suppressors are regulating the proto-oncogenes. The oncogenes are trying to like go forward. So it's not necessary that one single mutation might cause cancer. A lot of time, most of your cells have mutations to your proto-oncogenes or tumor suppressors. The key is when a cell kind of hits this tipping point where it's accumulated enough mutations to those proto-oncogenes and enough mutations to those tumor suppressors that it has a bunch of oncogenes and a bunch of non-functional tumor suppressors, that it reaches that tipping point where it can kind of just spin through the cell cycle, right? So whenever you're answering these questions, questions on your problem set, on your exam, things like that, make sure you keep that in mind, that a single mutation is not necessarily going to cause cancer, but it can, right? Any of these mutations can cause cancer, but that one alone probably won't. Does that make sense? Okay. So a couple more pro-oncogenes and tumor suppressors. We talked about P53 as a tumor suppressor. Now let's talk about an oncogene. That's E2F. E2F is a transcription factor in eukaryotes, and it controls the passage and moves forward through that G1 checkpoint, the restriction checkpoint, by acting as a transcription factor for genes like cyclin, CDKs, other checkpoint regulators, and DNA replication or repair machinery, right? So E2F is a proto-oncogene, because it is normally used for the growth of cells. It moves the cells forward through the restriction checkpoint from G1 to S phase, right? By acting as a transcription factor. So E2F is what goes and turns on all the other genes that are used in replication. It like goes around and wakes up the rest of the cell, and it's like, okay, it's time for replication. Let's go. Again, if something goes wrong, if you had this is a proto-oncogene, so if it becomes overactive, if you have a gain of function, it's going to constantly be waking up everything else. It's going to be constantly transcribing these other genes used in replication, and that's going to push the cell forward through the checkpoint, and it's going to constantly be pushing the cell forward in replication. Does that make sense? Yes? Okay. I'm just going to move forward. RB, on the other hand, is a tumor suppressor that functions as a regulator for E2F, right? We just talked about tumor suppressors being genes that regulate proto-oncogenes and prevent them from becoming oncogenes and preventing growth. RB directly regulates E2F, right? So what happens is it binds E2 when E2F is not supposed to be active, which is most of the time, right? It's only E2F is only supposed to be active when you're trying to actively pass that restriction checkpoint. So when the cell is just hanging out in G1 phase or even in G0 phase, the RB will bind to the E2F and like sequester it. If RB is bound to E2F, then E2F is inactive, right? So it's not able to carry out its function to push the cell forward through the cell cycle. So again, if something goes wrong, if you have something breaking, you have a loss of function in the tumor suppressor, the cell's can be able to slide by the checkpoint, because if you have a loss of function RB is no longer going to effectively bind and control E2F. So the E2F is in that, through that process, going to be able to carry out the E2F activity and push the cell forward. That's not necessarily a gain of function in E2F. That's a loss of function in RB that does that. E2F can have its own gain of function that pushes the cell forward through the cell cycle, but you can also have that essentially a similar effect by a loss of function in the thing that turns off E2F, right? Because in that scenario, you haven't mutated E2F at all. Actually, E2F is completely normal. It's just the regulation of E2F is now abnormal. Sounds good? Now I'm just going to spend like a half a second on this because I know you already went over this. This is the RTK MAP kinase pathway where a signal comes in. The RTK activates GRB2, which activates SOS, which activates RAS, which activates RAF, which activates MAC, which activates ERK. You don't even know the specific names of these, just know they're the MAP kinase, MAP kinase, and MAP kinase. And eventually through this like whole signaling cascade, you have activation of like proliferating cells. This is like cells that are proliferating genes, right? This is activating a survival pathway or survival signaling in the genes. So this pathway is a survival signaling pathway. It's just important to know that this pathway does that. So if you break anything in this pathway, now you're no longer signaling the cell to survive. So that might really lead to the cell now carrying out apoptosis. We don't go into any specific examples of any apoptotic pathways, but the opposite of this, where if you have some kind of pathway that results in activation of target genes that stimulate cell death, that would be called an apoptotic pathway. Right? It's not necessary that if you break something in this, it doesn't become an apoptotic pathway. It's just the survival pathway is not now off. So there's nothing actively telling the cell to survive. Sounds good? Okay. Then this is one last thing, is just like a semantics issue. I just want to make it clear. the difference between necessary and sufficient, right? So if things are necessary, then you need them to happen, but things that are necessary may not be sufficient, right? So multiple necessary mechanisms might be needed to make a sufficient mechanism, right? So sufficient is everything that you need, every mechanism that you need to achieve an end result, while necessary is each one of those steps that you absolutely have to have. And there's an example down here, which is that cyclin binding and CAC phosphorylation are both necessary for CDK to be activated, but both events together, or both events are only sufficient when they happen together to activate CDK. Because if you have just one of those, if you have just cyclin binding or just CAC phosphorylation, then the CDK won't be active. Okay. Now a couple more practice questions. What kind of pathway is the RTK MAP kinase pathway? And when I say this, I think I'm asking about proto-oncogene versus tumor suppressor. Would you say that the RTK MAP kinase pathway is a proto-oncogene or a tumor suppressor? Any thoughts? No thoughts?
Not the tumor suppressor one.
Yeah, exactly. Spot on. It is a proto-oncogene. Remember that the MAP kinase pathway, the RTK MAP kinase pathway, is a survival signaling pathway. So it's not telling the cell not to go forward. It's actually telling the cell to stay alive and stay moving through the cell cycle. If the MAP kinase pathway becomes overactive, it might tell the cell to stay alive even when the cell should not be alive. So if you have a tumor cell, a cancer cell, ideally you want the cell's own machinery to recognize that and be like, okay, this cell is replicating out of control. There's a lot of DNA damage. There's a lot of mutations. This cell needs to carry out apoptosis. But if you have this survival pathway running out of control, then it'll tell the cell to keep surviving despite the damage to the DNA and despite the negative things. And that would make this pathway an oncogene or an oncogenic pathway. So if RB has a loss of function, would this result in a cancer phenotype? To answer this question, first step is RB a proto-oncogene or is it a tumor suppressor?
Tumor suppressor.
Yes, it is a tumor suppressor. So if you have a loss of function in RB as a tumor suppressor, would this result in a cancer phenotype?
Yes.
Fantastic. Yes. The E2F would no longer be regulated. So the E2F will be able to act even when it's not necessarily supposed to, and you'd get some skipping past restriction checkpoint. It would just slide through, even if it should stop at that checkpoint. And now what happens if RB has a gain of function? Would this result in a cancer phenotype?
No, it would not.
Yes, exactly. It would not, and it would stall at the restriction checkpoint. This, depending on what's happening, this can actually also be bad. It wouldn't result in a cancer phenotype, but if this cell should be replicating and you have a gain of function in RB, then you might not have it replicating, right? Even though it should be. So even a gain of function in a tumor suppressor or a loss of function in proto-oncogene might still be bad. It won't necessarily result in cancer, but it might still be bad for the context of the cell and the context of the organism. Are CDKs proto-oncogenes? Are they considered proto-oncogenes? For this, think about what is the role of a CDK in the cell cycle and what do proto-oncogenes do in the cell cycle? Fair. So remember that CDKs are the cyclin-dependent kinases. They're the proteins that actually push the cell through the cell cycle. And if it's a proto-oncogene, they're used for the normal growth and replication of the cell, right? So if any protein relevant to the cell cycle and the cell's growth and proliferation, if that is causing the cell to move forward in the cell cycle, then it's considered a proto-oncogene. If it's causing the cell to stay where it is in the cell cycle, it's a tumor suppressor, right? Think about, is it the gas or is it the brakes? In this case, CDKs push the cell forward in the cell cycle, so they would be proto-oncogenes. That means that if you have a gain of function in a CDK encoding gene, you'd be pushing the gas too much, and the cell would skip through the cell cycle checkpoints and you'd have an oncogene. And I think it's the last question. If E2F will not let go of the DNA, is this a gain of function or a loss of function? Think about, what is the role of E2F? Like, how does E2F actually do its job? And what happens if E2F does not let go of the DNA? Excellent. So remember that E2F's function is as a transcription factor. So what it does is it attaches to the DNA, recruits RNA polymerase to come transcribe those genes, and those genes are all necessary for this cell to move forward, right? So regardless of what the function of those cells, those genes are, E2F's function is as a transcription factor. So the function of E2F is to attach to the DNA and bring in RNA polymerase. So if it does not ever let go of the DNA, that means once it's attached, it's staying on there. So it's constantly going to be bringing in RNA polymerase. This means that it's doing its job a lot better than it normally does, right? Regardless of whether the effect of that job is cancer, not cancer, good, bad, ugly, it's doing its job better. That's a gain of function. Sounds good? Well done.
Could we go over what a sufficient mechanism is again?
Yes. So sufficient mechanisms are enough for the intended result, right? I know it says here enough on their own. That's a little bit of a typo. It's just sufficient is whatever is like the bare minimum needed for the intended effect to occur. So if the intended effect is that you activate a CDK, right? You need two things to do that. You need cyclin to bind, and you need CAC phosphorylation, right? So only when those two things are both occurring is there a sufficient mechanism for CDK activation. However, in order for CDK to be activated, you need the cyclin to bind, and you also need the CAC to phosphorylate, right? So both of those things are necessary. So those two necessary mechanisms together make a sufficient mechanism. Sometimes you have mechanisms that aren't necessarily necessary, I guess, in which case they wouldn't be part of the sufficient part, because even if you don't need them, even if that doesn't happen, you might still get an intended effect, right? So if some mechanisms are not necessary, they're not part of what is sufficient for the end result. Okay, perfect. So I think it's all in terms of the slides and the questions. So if you guys have any questions, let us know. Do you know the answer to that?
I'm not sure, honestly.
Did she send an email?
I don't remember.
Do you have my phone?
Oh, I have my phone. Give me my phone. Unless she sent an email about that specifically, I don't remember if she did or not.
Okay, we'll
can
figure
check.
out the answer. Sky will figure out the answer to that. Do you guys have any other questions about the problem set, review sides, Laura's videos or lectures or anything like that?
I'm gonna look on Moodle. I don't see an email from her about it.
Wait, did something prompt you to ask that did she say something about that? Do you want to end the recording now?
Oh yeah.
We're just gonna end the recording real
Wait.
quick.
Where's the recording button?
Were you recording?
Oh, yes.